In this video, we'll be covering the second part of Chapter 5, Chemicals and Cancer. We had mentioned previously that there are a number of chemical carcinogens and that these can be grouped into a number of different categories. And we had kind of discussed how they are studied. How do we know that they cause cancer? What dose do we know that they cause cancer with? In some cases, carcinogens need to be processed by enzymes, typically in the liver. And then once that process happens, then they have their true carcinogenic ability inside of our bodies. In the liver, we have an enzyme called cytochrome P450. And this enzyme is known to oxidize foreign chemicals. And the goal would be to make them less toxic and also easier for them to be excreted from the body. So this reaction overall that is catalyzed by cytochrome P450 makes that chemical more water soluble, and thus it's able to be excreted in the urine. However, sometimes this reaction can convert a pre-carcinogen into a carcinogen and lead to carcinogen activation. So here is cytochrome P450, and it has the ability to bind to chemicals. Cytochrome P450 contains an iron inside of it, and this iron is reactive. So what happens, you have step 1, you have the substance binding to cytochrome P450. Step 2, an iron atom in cytochrome P450, so it's part of the enzyme group, is reduced by an electron. So we see the electron coming in. We see the charge changing from a positive 3 to a positive 2 charge, right? Here we have oxygen coming in. Oxygen can bind to this iron atom. And then we have a reaction occurring. So the reaction occurs with another electron and 2 protons, and this leads to the production of a water molecule. And then the last reaction is that the substance being oxidized is hydroxylated and released. So the beginning product and the end product are the same, but there are many steps in between, right? And the overall process results in hydroxylation of the chemical. And so this would make the chemical more water soluble and more likely to be excreted from the urine. However, sometimes this creates a carcinogen from a pre-carcinogen. Levels of cytochrome P450 have been studied in individuals that have higher exposure to chemicals like tobacco smoke. And then if a person is exposed to higher levels of carcinogens, it's actually noted that they have an increased level of cytochrome P450. If you remember in the Ames test, to do that test, the chemicals were incubated with liver extract, which would include this enzyme. And so that enzyme would have the ability to do this reaction before that carcinogen is tested in the Ames model. When we think about chemicals being carcinogens, we should recognize that carcinogens are typically molecules that like to undergo different reactions. They're not just inert. And in many cases, carcinogens would be electrophilic. So that term electrophilic means electron loving. So the more electron loving a molecule is, then the more carcinogenic it's likely to be. So these chemicals like to react with other molecules that are rich in electrons, possibly taking those electrons away from them. And so some of these molecules include DNA, RNA, and protein. So looking at various chemicals, this experiment is showing us that when we look at these chemicals and we look at their ability to bind to and react with DNA, so here we have the increased ability to bind to DNA, and then here we have their level of carcinogenic potency. So their ability to bind to DNA and react with DNA directly correlates with their ability to act as a carcinogen. In this slide, we're taking a closer look at benzoate pyrene, which is a known carcinogen. When this molecule enters the body, it is then going to be activated by cytochrome P450. So the reactions shown here are a series of reactions that occur by cytochrome P450, and in each reaction we see that the molecule is becoming more and more water soluble. So here's the original molecule. Here's a reaction that adds an epoxide group. Another reaction, here we have two OH groups. Here we have a reaction that has epoxide plus the hydroxyl groups, and here we have 4 of those hydroxyl groups. So we have increasing water solubility with this molecule. But one of the reactions along the way has the ability to undergo reactions with DNA. And so that's why we see the arrow over here indicating that it can interact with the DNA strand and specifically interact with the nucleotides on that strand. So it's showing you guanine and how guanine can react with this molecule. When we have them forming a bond, what occurs is that you have distortion of that double-stranded piece of DNA. When you have distortions of the double helix, then that can result in mutations being kind of put into that piece of DNA strand during the DNA replication process. Because as DNA polymerase comes up to this strand and it's trying to add in the new nucleotide, then it's going to see this distortion, and so it might put in the wrong base at that spot. So epoxide formation is known to create this problem, and this is what we call DNA adducts. This is when we have a DNA and a carcinogen that form a complex. It distorts the DNA double helix, which results in mutation during replication. So this can occur for a number of different carcinogens, and it also includes aflatoxin, vinyl chloride, as we had in our example in class. So there are many sites within the DNA bases that can be targets for these carcinogen attachments. And there can also be other electrophilic groups made based off of other ions and also free radicals. So in our diagram here, we have all of these different locations on our various bases that could be reactive spots for these electrophilic molecules. Overall, the ability to form DNA adducts is one of the best predictors of a molecule's capacity to cause cancer. And that is because they can form the DNA adducts, which results in disruption of the double-stranded DNA helix, and that can lead to mutations, and then the mutations are in the end what causes cancer. So we gave you the example of the carcinogen forming epoxides, and then ending up with this distortion of the DNA strand. But there are some other things that can happen as well. So, and there's a lot of variability here. You could have a single strand break, so we're just breaking one side of the DNA. We could have a link between adjacent bases. We could have a cross link between the DNA strand bases. We could have a full double-stranded DNA break. We could have hydroxylated base, a missing base, and then, as we mentioned before, a DNA adduct. When we talk about the development of cancer, we often mention that there is an initiation step and a promotion phase, or step. So two major phases here. So in this animal model that's being shown here would be animals that are treated with a carcinogen and looking at their ability to form tumors. So in this top graph, we have an animal that was treated in one dose with the carcinogen, the MPA, and then they were not treated again. And then the animals followed over time, and they did not form the tumors. In the second model, we have that initiation event, the exposure to the carcinogen, and that was a one-time exposure. And then some time went by, and then they were exposed to croton oil. And that allowed for the development of tumors to be made. Croton oil is extracted from the croton plant, but it is a poisonous liquid that causes irritation. So we would have long-term exposure to this agent that causes irritation. And they treated it 2 times per week. And what they saw is they saw an increased rate of cancer. Now, if they only treat with croton oil, they do not produce skin tumors. So this model is giving us evidence here that we have 2 main steps here. One is the tumor initiation event, where we have treatment with a carcinogen. So we have a one-time exposure to a carcinogen. And then 2, we have tumor promotion. We have something that causes long-term inflammation at a certain site, and this is allowing for a promoting event. So allowing for that tumor to actually grow. When we talk about tumor initiation, we are typically talking about exposure to some sort of carcinogen, oftentimes genotoxic. So here is a chemical or other carcinogen that is something, an event, we are exposed to it, it's causing DNA damage. And that DNA damage is going to lead to mutations. And it's the mutations which may or may not cause cancer. So here we have an example of a mutation that occurs on DNA. So we had a G and a C, then we have exposure to this agent, and this agent is able to add a methyl group to the guanine base. Now that we have this methyl group here, the cell is going to, at some point, undergo DNA replication. And as it's replicating the double-stranded piece of DNA, as it replicates this side, then it correctly puts the right base. So G and C, that is correct. There is no mutation from that replication event. Over here, when it's replicating the other side of the DNA double helix, it's going to look at this methylated guanine, and then it has to put in a nucleotide. So it looks at it and it thinks like, oh, this is actually an adenine and I need to put a thymine there. And then when we get to go the second round of DNA replication, let's say the cell divides again, then this is going to be further embedded into the sequence. So here we have permanent mutations over time, where we go from having a GC base pairing to an AT base pairing. So this is our mutation event, and then after DNA replication, it becomes kind of like a permanent mutation that continues to be passed down from as the cell is doing DNA replication. Now the tumor that could arise from this, it's not going to occur right away. It would occur many years after this mutation event. And also keep in mind this mutation event is random, so we don't know exactly like what gene or exactly where in the DNA this mutation occurs. So only if the mutation occurs in a specific type of gene or even in the specific area of a gene would that mutation cause an actual event that could lead to cancer. When we think about tumor promotion, this is a longer time process. It's a gradual process. We have prolonged, repeated exposure to a promoting agent. And the agent typically stimulates cell proliferation. So it allows for the cells to keep dividing, and that is going to allow for a situation where we can ultimately develop cancer. So as we mentioned, promoting agents typically stimulate cellular proliferation. And sometimes they do that by leading to activation of various cellular pathways that ultimately increase cellular proliferation. So in this model shown here, we're looking at formyl esters. And they have the ability to act as a tumor promoter because they can stimulate cell proliferation. And this diagram is showing us how they do that through the activation of protein kinase C. So in the normal situation, if we're going to have activation of this pathway, we would require an external signal to bind to a receptor that's going to lead to activation of a cell signaling pathway. And so in the first step here, we have activation of phospholipase C. When phospholipase C is active, it will cleave the membrane part, PIP2, and it produces the diacylglyceride, which remains in the lipid. Diacylglyceride, DAG, is going to activate protein kinase C. Protein kinase C will lead to a series of activation events, which ultimately leads to cellular proliferation. Now, where does formyl ester come in? So formyl ester is a molecule that can mimic the action of DAG here. And so it alone can activate protein kinase C. So it is going to activate cellular proliferation in the absence and not depending on the receptor. So this could lead to sustained cellular proliferation over a long time. Here we have TPA, a commonly known tumor promoter. So this is a chemical that is a formyl ester, and it also has the ability to activate PKC, as we just mentioned. Looking at different chemical modifications of this molecule, there can be small changes made in the structure that can allow this chemical to be used but have less ability to promote tumorigenesis. So sometimes when it comes to regulating these chemicals or looking at your exposure to them, you could possibly look at the chemical and what are some variations on that chemical that could be used that would have, less properties to be a tumor promoter. So in this case, less ability to activate PKC. There are other molecules that are known to activate PKC, and this includes a fungal toxin and an algal toxin that are listed here. So in general, I know a lot of times when we think about tumor initiators and tumor promoters, we think that these are going to be things that are happening outside of our body that we are exposed to. But it is possible for us to have natural tumor promoters. So not all tumor promoters have to be a foreign substance. So remember, tumor promotion is all about just increasing cellular proliferation. That's it. So there are other things that we have that can increase tumor promotion. So this includes hormones. So estrogen and testosterone, both of those can stimulate cellular proliferation and then can lead to increased rates of breast, ovarian, and prostate cancer. Other things that we are exposed to, like dietary fat, our diet, things like that, can act like tumor promoters. And sometimes you hear people talking about inflammation. If long-term inflammation can also result in this tumor promoter kind of process too. So we've mentioned that we need tumor initiation and tumor promotion in order for us to be going along the road of carcinogenesis. And what we haven't mentioned yet is progression. So we have step 1, initiation. That's our exposure to our carcinogen, something that causes a mutation. Step 2, our exposure to something that's going to cause cells to proliferate faster than they would without the mutation. And this process can result in increased ability to obtain other mutations and also other epigenetic changes. And so as these cells proliferate rapidly, they have the ability to kind of like evolve and change over time. Step 3 is progression. So progression is the gradual change in the property of our tumor cell population. And so we are kind of indicating here, and we had mentioned our different clonal populations in a previous chapter, where we can have a mass of cells growing that start to undergo different mutation or genetic changes, or epigenetic changes, I meant to say, and that can lead to different populations of the cell being present. And so here we have the 3 colors of cells being represented here. So these are all derived from the same cell, but they've undergone different genetic mutations. And so over time, we have a process of proliferation and selection, and eventually the most aggressive cell will be selected for, and then we will have clonal selection, which is just going to represent a clone that is the most successful at high proliferation and ability to survive. So one question could be, well, what is the source? What's really causing a cancer cell to be a cancer cell? Is it DNA? Is it RNA? Is it protein? Is it something else? And so these questions can kind of be answered through various studies. And in this case, what they're doing is they are taking melanoma cells and they're asking the question, if I just move the DNA from that cell into another cell, does just the DNA by itself have the ability to develop cancer? So what they're doing is they take an egg cell, so this is a stem cell, right? An egg cell. They remove its nucleus. They replace it with the nucleus from the melanoma cells. And then they allow it to start to grow and divide and start to form an embryo. Here they remove stem cells from here. So these stem cells would contain the nucleus from the melanoma cell, and they grow them in a dish. Then they take these stem cells and they inject them into a normal embryo. They implant the embryo into a normal mouse and allow it to develop into a grown mouse. And what they see is that these cells were part of the process of embryogenesis, so they end up in various places throughout the body, and only those cells contain these cancer cell nuclei. This experiment gives evidence that it is the DNA in the nucleus that is the cancer causing agent in the end. And then we kind of make a note that while we are very familiar with mutations being linked to development of cancer, there can be also epigenetic changes, so alterations on top of DNA, such as methylation and things like that, that can lead to alterations in gene expression and can lead to the development of cancer. So to kind of summarize, if an agent is given the name a carcinogen, that means that that molecule has the ability to increase the probability of development of cancer. This will not happen in every single case. It is a little bit by chance. It depends on if the mutation is made, where in the genome is it made, and that kind of leads to why we have such variability here. It's going to depend on the dose. So higher dose is higher risk of cancer. The potency. So different chemicals have different abilities to cause like DNA adducts or changes on DNA. And this leads to us saying things like we have altering levels of carcinogenic potential. We have our graph here. We have various carcinogens, and then we have different doses at which they are listed as a carcinogen and have the ability to result in the development of cancer. We also have a couple of categories: incomplete carcinogens. These carcinogens act as a initiating or promoting agent. The key word is or. It does one or the other, but it does not have the full capacity to develop cancer all by itself. Complete carcinogens act as the initiating and promoting agent. Their ability to act as a complete carcinogen is often dose dependent. And an example of something that is a complete carcinogen would be like the tobacco smoke or coal tar, which have a mixture of chemicals. And so they have this mixture of chemicals that can act as initiating and promoting events. If we're talking about cancer potency, so how tumorigenic or carcinogenic is a molecule, this can be influenced by a few things: the electrophilic strength of the carcinogens, and then its ability to be activated by cytochrome P450. So we've studied some of the properties of these chemicals, why some of them are carcinogenic, and also how they become carcinogenic, and then how they cause cancer, right? not every carcinogen has the same amount of potency as we saw in the graph before. And not every single exposure to a carcinogen is going to lead to the development of cancer. We mentioned that if they do make a mutation in the DNA, that mutation occurs randomly, and it could occur anywhere in the genome. So it could occur in a spot where it doesn't really make a difference. However, if it occurs on a gene like P53 or retinoblastoma, then now that mutation is kind of residing inside of a critical gene. And as we talked about in previous chapters, mutations in those genes have a high rate of causing cancer. And then again, we mentioned previously, the potency can vary widely. This can be due to whether or not it's activated by cytochrome P450, its electrophilic strength, how strongly does it pull electrons off of other molecules, how reactive is that chemical with DNA, how likely is it to create mutations. So all of these factors kind of come into play with whether or not a compound is carcinogenic, and then again, whether or not the person exposed to that compound would actually develop cancer. There are other factors that can be taken into place here also, genetic risk factors. Some people have other things kind of going on that can cause them to be more likely to develop cancer. And then also, if a person is taking immunosuppressive drugs, that can definitely contribute to their cancer development because their immune system is not detecting abnormal cells and clearing those out. So it does result in a higher risk of being able to develop cancer. So that ends our video for Chapter 5, reviewing how do we know if a chemical causes cancer, what kinds of studies need to be done, what are the properties of molecules that cause them to cause cancer, what step are they involved in, the initiation, mutation step, or the promoting step, and then also just talking about this random nature of the development of cancer. It really is sometimes just by chance. Thank you for listening.